day everyone! Today, I'm here to present the simulation of the different stages of mitosis and meiosis. But before we proceed, let me first introduce the different parts of animal cell, which is very important to remember as we come up simulation activity. This is an example of an animal cell with different parts. First, we have a cell membrane, the centrioles, the nucleus, and the nuclear envelope. This color green is the nucleolus, while this red is the maternal chromosome and blue is for paternal chromosome. Let us proceed to the different parts of mitosis. Divided cells undergo irregular patterns or of events. This cycle may be divided into two basic parts wherein the first stage of mitosis, which is interphase, the cell undergoes a period of intense chemical activity and the amount of DNA is doubled during this period. Interphase is divided into three parts. The G1, or first growth phase, is where cell division and most of the period of cell organelles are synthesized and cells grow rapidly. Next is the synthesis or S phase, where in this phase the DNA replication occurs. And lastly, the G2, or second growth phase, is where centrioles replicate and energy stores increase. Let us proceed to the second stage of mitosis, which is prophase. In this stage, the chromosomes become visible as dawn. The chromatin gradually coils into chromosomes, which is compound of two chromatids bound with centromeres. The centriole migrate to the opposite ends of poles of the cell. From each centrioles, the microtubules develop, forming a star-shaped structure called aster. Now, the nucleolus disappears and the nuclear envelope disintegrates. Now the chromosomes are free-floating within the cytoplasm of the cell. Next is the third stage of mitosis, which is metaphase. The chromosomes arrange themselves at the center or equator of the spindle and become attached to certain spindle fibers at the centromere. Centromere of chromosomes connect to the spindle fiber. Next stage of mitosis is anaphase. The centromeres split and further shortening of the spindle fibers causes the two chromatids of each chromosome to separate and migrate to opposite poles. The shortening of the spindle fibers is due to the progressive removal of the tubulin molecules of which they are made. The energy of this process is provided by mitochondria, which are observed to collect around the spindle fibers. For the last stage of mitosis, we have telophase. The chromatids reach their respective poles and a new nuclear envelope forms around each group. the chromatids uncoil and lengthen, thus becoming visible again. The spindle fibers disintegrate and the nucleolus reforms in each new nucleus. Now that we are done in the simulation of the different stages of mitosis, let us proceed to the simulation of the different stages of meiosis. Meiosis involves one division of the chromosomes followed by two divisions of nucleus and cell. The result is that the number of chromosomes in each cell is reduced by half. The diploid parent cell gives rise to four haploid daughter cells. Meiosis occurs in the formation of gametes, sperm, and ova in animal cells. Let's come up to the different stage of meiosis. First is the interphase. In interphase, the cell is in the non-dividing condition during which it replicates its DNA and organelles. Next is prophase 1. In prophase 1, organisms have two sets of chromosomes, one derived from each parent. Any two chromosomes which determine the same characteristics are called an homologous pair. Although each chromosome of a pair determines the same characteristics, they need not be identical. Prophase 1 of meiosis is similar to prophase in mitosis, in that the chromosomes become visible, shorten and fatten, but differs that they associate in their homologous pairs. They come together by a process termed synapses, and each pair is called a bivalent. In early prophase 1, we have centrioles, the nucleolus, nuclear envelope, chromosome with a varied appearance due to chromomeres, the cell membrane, and the asters. While in the mid-prophase, the nucleolus disappear, and the chromosomes come together in their homologous pairs. The centrioles will remain and the nuclear envelope. While in the late prophase 1, the spindle fiber are visible. The nuclear envelope disintegrates, and each chromosome is seen to comprise two chromatids. This is the point of crossing over of the chromosomes. The next stage of meiosis is metaphase 1. In metaphase 1, the bivalents arrange themselves on the equator of the cell with each of a pair of homologous chromosomes orientated to opposite poles. Centromeres are pulled towards the poles. And this is now the equator. This arrangement is completely random relative to the orientation of other bivalents. The spindle fibers attached to the centromeres contract slightly, pulling the chromosomes apart as much as the chiasmata allow. The next stage of meiosis, we have anaphase 1. In anaphase 1, the spindle fibers which are attached to the centromeres contract and pull the homologous chromosomes apart. One of each pair is pulled to the one pole and its sister chromosome to the opposite one. This is one of the homologous pairs of chromosomes being pulled to a pole. This piece of chromatid from sister homologous chromosomes are exchanged during crossover in prophase 1. Now, let us proceed to the next stage of meiosis, which is telophase 1. In telophase 1, the chromosomes reach their opposite poles and the nuclear envelope forms around each group. In most cells, the spindle fibers disappear and the chromatids uncoil. In this stage, the nuclear envelope reforms, while the nucleolus is also reforming. And this is the cell dividing by constriction. Let us now proceed to prophase 2. In those cells where telophase and interphase take place, the nucleolus disappears and the nuclear envelope breaks down. Where the centrioles are present, this divide and move to opposite poles. The poles on this occasion are at right angles to the plane of the previous cell division, and therefore the spindle fibers develop at right angles to the spindle axis of the first meiotic division. In this stage, which is prophase 2, the spindle fibers forming at right angles to previous spindle axis. The nuclear envelope disintegrating. Next is the metaphase 2. The chromosomes arrange themselves on the equator of the new spindle. The spindle fibers attach to the centromer of each chromosome. In metaphase 2, chromosomes lying at the equator of the spindle. Next is the anaphase 2. The centromers divide and are pulled by the spindle fibers to opposite poles, carrying the chromatids with them. In anaphase 2, the chromatid moving to pole. So this is the chromatid. For the last stage of meiosis, we have telophase 2. The telophase 2, upon reaching their opposite poles, the chromatids unwide and become indistinct. The nuclear envelope and the nucleolus are reformed. The spindle disappears and the cells divide to give 4 cells, collectively called a tetrad. In telophase 2, the tetrad of 4 cells is formed. Each cell has a haploid number of chromosomes. Crossing over during prophase 1 has produced genetic variety, which in 6 example has given 4 cells of different genetic composition. And that's the end of the simulation of different stages of meiosis and mitosis.